When science tried to reconnect the spinal cord for decades, spinal cord injury represented a boundary. When neurons in the medulla were severed, the message was clear. Communication had been interrupted. Signals from the brain could no longer reach muscles below the lesion. Movement stopped. Sensation faded. The body, once responsive, became silent. Medicine could manage symptoms. It could stabilize. It could adapt, but it could not reconnect. That limitation shaped how paralysis was understood. Then research in Brazil began to challenge this boundary. At the Instituto de Ciências Biomédicas, the UFRJ biologist and researcher Tatiana Coelho de Sampaio dedicated more than 2 decades to studying extracellular matrix proteins and their role in neural regeneration. The result of that research was a molecule called polilaminina. To understand why this matters, we need to understand what fails in spinal injury. Neurons do not simply disconnect like wires. When the spinal cord is damaged, scar tissue forms. Inhibitory molecules accumulate. The local environment becomes hostile to regeneration. Axons, the long projections of neurons, attempt to regrow, but they lack structural guidance. They do not know where to reconnect. The nervous system, unlike peripheral nerves, regenerates poorly in the central nervous system. The problem is not only damage, it is the environment. Polilaminina was developed to modify that environment. Laminin is a natural protein found in the extracellular matrix. It plays a role in cell adhesion, differentiation, and migration. It provides structural scaffolding that cells use to orient themselves. Tatiana Sampaio's work focused on creating a polymerized form of laminin, polilaminina, capable of forming a structured network at the site of injury. Not a drug, not a stimulant, a scaffold. The idea was not to force neurons to grow. It was to provide physical and biochemical orientation so that axons could reconnect across the lesion site. In physical terms, this is structural guidance. Neurons extend processes along gradients. They respond to molecular cues. Growth cones at the tips of axons navigate using chemical and mechanical signals. If the environment lacks structure, regeneration stalls. Polilaminina creates a mesh-like matrix that acts as a biological bridge. It reduces inhibitory signals and provides adhesion sites for axonal growth. This is not mystical regeneration. It is guided reorganization. Experimental applications in patients with spinal cord injury showed promising results. At least 6 patients with paraplegia or tetraplegia demonstrated partial recovery of sensation and voluntary movement following surgical application of the molecule. One documented case, Luis Fernando Moser, previously exhibited regained sensitivity and partial muscular control in the thighs. These results are significant, but they are not definitive. The treatment remains in experimental stages. According to public reports, the research is conducted in partnership with the pharmaceutical company Cristália, and regulatory review by Anvisa is ongoing, with additional preclinical data requested. This is important. Science does not declare victory early. The nervous system is complex. Regeneration is variable. Long-term outcomes must be validated. Controlled trials must confirm efficacy beyond isolated cases. But the principle revealed by this research is profound. The spinal cord may not be as irreversibly static as once believed. Biologically, regeneration depends on microenvironment. Physically, Growth depends on structure. Neurons are not passive. They attempt to reconnect. But without scaffolding, growth is chaotic and often abortive. With structured guidance, reconnection becomes statistically more probable. This aligns with fundamental principles of physics and biology. Complex systems reorganize when given stable constraints. Cells follow gradients. Axons follow adhesion. Growth follows structure. The success of polilaminina suggests that regeneration is not solely limited by intrinsic cellular inability. It is limited by environmental architecture. Modify the architecture, and new possibilities emerge. This connects biology directly to physical law. Proteins form structures through molecular bonding governed by thermodynamics. Adhesion depends on electrochemical interactions. Signal propagation depends on ion channel dynamics. None of this violates physics. It expresses it. Regeneration is not a miracle. It is matter responding to constraint. For decades, central nervous system injury was treated as permanent because the inhibitory environment overwhelmed regenerative capacity. Tatiana Sampaio's work attempts to rebalance that environment, not by overriding biology, but by collaborating with it. This represents a shift in medical thinking. Instead of stimulating neurons aggressively, the approach modifies the matrix around them. It provides structural continuity where discontinuity once dominated. The difference is subtle but powerful. Life does not need to be forced to heal. It often needs the right geometry. The broader implication extends beyond spinal injury. If structural guidance can restore partial function in the spinal cord, similar principles may apply to other neurological injuries: stroke, traumatic brain injury, degenerative disorders. But caution remains essential. The results are promising, not universal. Clinical validation is ongoing. Regulatory approval requires rigorous data. Scientific credibility depends on reproducibility. Hope must remain aligned with evidence. Yet, even at this stage, something important has shifted. The idea that central nervous system damage is categorically irreversible has weakened. Biology appears more plastic than assumed, and plasticity is not magic. It is organized adaptation within physical constraints. Neurons are matter. Proteins are matter. Growth is matter. But matter arranged correctly becomes recovery. The work at UFRJ does not overturn physics. It confirms it. Structure enables function. Modify structure, and function can change. In the quiet laboratory environment where polilaminina was developed over 25 years, what emerged was not simply a molecule. It was a demonstration that regeneration may depend less on force and more on form. The spinal cord is not only damaged tissue, it is a system awaiting structural permission. Science is now asking whether that permission can be granted. Still, within